Good morning, Ms. Jin. Yes. Hereby I open this academic ceremony in which Ms. Shunxin Jin will defend the academic thesis, The Alternative Pathway of the Complement System in Vascular Comorbidities of Obesity and Type 2 Diabetes. May I invite you to present a summary of your studies in the next 15 minutes and basically also the conclusion of your thesis. I'd like to give you the word.
Thanks, Prorector. I will present my thesis. Dear Prorector, dear members of Corona, dear colleagues and friends on-site and online, good morning, everyone. Thanks for coming my PhD defense ceremony. In the next 15 minutes, I will present my thesis titled The Alternative Pathway of the Complement System in Vascular Comorbidities of Obesity and Type 2 Diabetes. Nowadays, obesity is very common all over the world. It is easy to find obesity and overweight on the street. People may ask, what is overweight and obesity? Overweight and obesity are defined as abnormal or excessive fat accumulation that may impair health. Since 1975, obesity is getting more and more. Worldwide presence of obesity almost tripled since 1975. Around 40% adults are overweight or obese in 2016. What will obesity bring to people? Obesity will bring people abnormal blood lipid level, high blood sugar, cardiovascular disease, and type 2 diabetes. Obesity may bring one people one of these diseases, or obesity may bring one people all of these diseases. Cardiovascular disease is the major cause of death. Cardiovascular disease is one low-grade inflammatory disease of the vascular wall. The major cause for cardiovascular disease is atherosclerosis. Atherosclerosis is lipid accumulation in blood vessel wall. The yellow part of this cross-sectional blood vessel wall is atherosclerosis. Atherosclerosis happens when the inner lining of a vessel wall damaged. Another reason for cardiovascular disease is stiff blood vessel. Compared to normal blood vessel, stiff blood vessel has less flexibility. Type 2 diabetes is another disease that obesity brings to people. Obese people more likely to have type 2 diabetes than lean people. People with type 2 diabetes are more likely to have cardiovascular disease. People may ask, in what condition people will have type 2 diabetes? If one people have insulin resistance and do not have enough insulin secretion, people will have type 2 diabetes. Insulin is produced by our body organ, pancreas. Insulin could lower blood glucose level. Insulin resistance means insulin can't work well. It can't lower our blood glucose level. Then we will have higher blood glucose level. Then people may have type 2 diabetes. Complement system is a group of proteins. It is one part of immune system. It is our bodyguard. It can kill bacteria and protect us from bacteria hurting. Complement could be produced in fat tissue. Complement could also be produced in liver. It also can be produced in blood vessel. After production, complement was transported to bloodstream. It can work for blood vessel and homeostasis. If we have enough complement, we keep healthy. If we have less complement than normal level, we will be hurted by bacteria. If we have more complement level than normal level, what will happen? We also can be hurted. Nowadays, some publications show that complement activation may involve in cardiovascular disease and type 2 diabetes. Complement has 3 pathways. They are classical pathway, lectin pathway, and the alternative pathway. In this thesis, I focus on the alternative complement pathway. In alternative complement pathway, I focus on 3 complement factors. They are complement C3, complement factor D, and complement C3A. Complement C3 is one key factor of the alternative complement pathway. Factor D is one enzyme. It could cut complement C3 into C3A. In this thesis, there are 3 study aims. Aim 1, to study the association between complement and cardiovascular disease. Aim 2, we study the association between complement and type 2 diabetes. In Aim 3, we study the effect of weight loss on complement system concentration. Data of Chapter 2, Chapter 3, and Chapter 4 were from the Maastricht study. This study had around 9,000 participants. These 9,000 participants are from the southern part of the Maastricht of the Netherlands. Maastricht study is one observational study. Observational study is one kind of study that we can't interfere participants' lifestyle or habits. We just can record or measure their characteristics and then analyze them. In this thesis, I used part of data from Maastricht study. It has around 3,000 participants. Half of them are men, half of them are women. The first aim of this thesis is to study the potential association between complement concentration and cardiovascular disease. To study this association, we have several outcomes. They are low-grade inflammation, damage of inner lining of a vessel wall, the thickness between inner lining of a vessel wall and middle layer of blood vessel wall. Another outcome is blood stiffening. The last outcome is cardiovascular disease. In this analysis, we found more complement concentration are associated with more low-grade inflammation. We also found more complement are associated with damage of inner lining of a vessel wall. We do not find more complement concentration associated with thickness of inner lining of a vessel wall and middle layer of a vessel wall. We also do not find complement concentration associated with blood stiffening. We found people with more complement concentration are more likely to have cardiovascular disease. Obese people more likely to have type 2 diabetes. Therefore, the second aim of this thesis is to study if the association between obesity and type 2 diabetes, or part of the association between obesity and type 2 diabetes could be via complement factors. We found obese people had more complement concentration. People with more complement concentration have more likely to have type 2 diabetes. Obese people are more likely to have type 2 diabetes. Part of the association between obesity and type 2 diabetes could be explained by complement factors. Based on the result from Chapter 2, Chapter 3, and Chapter 4, the third aim of this thesis is to study the effect of weight loss on complement concentration in Chapter 5. Data of Chapter 5 was from weight loss study. This study has around 50 obese men. They were divided into weight stable group and weight loss group. Half people in weight stable group, half people in weight loss group. People in weight stable group, they keep their previous diet. People in weight loss group, they had a very low calorie diet to lose their weight. The result for this analysis, we found weight loss could reduce complement concentration. The reduction of complement concentration could improve the damage in the lining of a vessel wall. We also found weight loss could improve the damage in the lining vessel wall. Part of the effect of weight loss on improving damage in the lining of a vessel wall could be explained by reduction of complement concentration. In conclusion, more complement concentration were associated with low-grade inflammation, with vascular damage, and also associated with cardiovascular disease. More complement concentration associated with type 2 diabetes. We do not find more complement concentration associated with blood stiffening. We found weight loss reduce complement concentration and improve blood vessel wall damage. This thesis may provide a clue for prediction, prevention, and treatment for cardiovascular disease. However, all possibilities need to be further studied. Thanks for your attention. Now I will give words back to Prorector.
Thank you very much, Ms. Candidate, dear Ms. Jin, for your very clear presentation, well illustrated, and it gives the opportunity now to start the discussion. And the opposition will be opened by Professor Rennenberg. He is affiliated with the Department of Promotion of Quality and Safety in Healthcare, and he was also, a member of the assessment committee, and I'd like to give him the first word.
Thank you, Prorector. Dear candidate, first, my congratulations with your fine thesis, and I would like to extend my congratulations to your promotional team. I read it with great interest because there's also in it about atherosclerosis and complement. That was my interest many years ago, and I wrote my own thesis relating to that subject. And so I started reading in chapter 1. Most people do when they open a book, start reading in chapter 1. And I saw at page 16 in chapter 1 that you investigate factor D, which leads to beta cell preservation. Chapter 1 is not your own investigation, but it's just a summary of the literature. And factor D and beta cell preservation, that's interesting because beta cell preservation might lead to less diabetes. although if we read on and go to page 39, table 2, we can see that even if you correct for type 2 diabetes, factor D still is associated with more inflammation, which should lead to more diabetes, more inflammation, more diabetes. Actually, more inflammation, the beta cell dysfunction or beta cell decline usually is also due to more inflammation. So, although the relationship is stronger without correction for factor D, one might suspect it protects people from getting type 2 diabetes. So, can you elaborate on what the physiological thought is here? Is glucose mightier than factor D in damaging endothelial function or beta cells?
Thanks, highly esteemed opponent. Thanks for your question. I didn't get it very well. Could you please repeat your question?
Yeah, so I was wondering, factor D is associated with beta cell preservation. You state that in chapter 1. There's literature about that. And then there is a strong association with inflammation. So if factor D goes up, inflammation goes up. So what you showed us in your slides. And then we expect it to lead to more diabetes. And but then further on, I read that factor D also might protect people from getting type 2 diabetes because of beta cell preservation. I think that's a little bit contradictory.
Highly esteemed opponent, thanks for your question. Yes, as what you said, we do find complement factor D positively associated with inflammation. If we just, according to this, we may think complement may contribute to type 2 diabetes. And for this association, for this positive association between complement factor D and inflammation, it can work on the insulin resistance. We found in the chapter 4, we found the complement factor D positively associated with insulin resistance. It may be via inflammation. And why complement factor D can protect people for type 2 diabetes? We think because complement factor D could work on the beta cell function and the beta cell survival, and for that reason, then people could have more insulin secretion, and the people will have less fast blood glucose, and then they will have a protective factor D may have a protective effect on type 2 diabetes.
Yeah, so actually it's both ways. So factor D does induce beta cell preservation, and on the other hand it induces inflammation. And could you elaborate on why nature thinks about something like that? So first it makes things worse, and then it does something to make things better.
Thanks for your question. Yeah, this is a very interesting question. Yeah, and for factor D is from the alternative pathway, and for the complement they have 3 pathways, and also for the other 2 pathways they have the similar situation. The one side they have the protective effect for us, and one side they do the harmful things for us. I think maybe they just want to keep balance.
Yeah, the body wants to keep balance. I always thought it's just in the reproductive years that it is made to do that, and your people in your studies are a little bit older, I guess.
Yeah, they are not very young. Yeah. They are not very young. They are the age of them is between 40 and 75. They are not very young, and they are oversampled with type 2 diabetes compared with normal population, they have more people with type 2 diabetes and have more older people.
Yeah. I would like to ask you something about chapter 2. On page 45, you state that the risk for atherothrombosis is higher than the risk for atherosclerosis, although low-grade inflammation might also generate atherosclerosis. That's what I found in my studies. It's a long time ago, so maybe these are very new findings and I have to adjust. But could you explain this difference? Because if there is continuously low-grade inflammation, I would expect atherosclerosis, and it was not there. Has it anything to do with your population, the choice of your population, you think?
Yeah, in the chapter 2, we do found complement factor D are positively associated with low-grade inflammation and endothelial dysfunction, and we do not find they are positively associated with carotid intermediate thickness and ABI, ankle brachial index, and we think they are not work on atherosclerosis. Maybe complement factor D maybe via inflammation and endothelial dysfunction, then via the atherothrombosis. Yeah.
Yeah. Thank you for your answers. I give the word back to the prorector.
Thank you very much. The opposition will now be continued by Professor Van Dijk. He is affiliated with the Department of Genetics and Systems Biology of the Metabolic Syndrome at Leiden University. And of course, I would like to thank him that he joins this morning, and I'd like to give him the word. Professor Van Dijk.
Thank you, Director. Dear candidates, first of all, I would like to congratulate you with the completion of your thesis. And I would also like to take the opportunity to congratulate your promotion team with this accomplishment. I also enjoyed reading your manuscript and learn about the alternative complement system. And since I'm also interested in the role of adipose tissue in the pathogenesis of disease, in the past, we've also studied gene expression in both visceral and subcutaneous adipose tissue of humans, and we found that complement factor C3 is really one of these genes that's very highly expressed, and that's always puzzled me. In our studies, it wasn't a differentially expressed gene, so we didn't focus on it, so I didn't really look into the biological meaning of this high expression, and that's what I would like to ask you. So, dear candidates, could you explain why adipose tissue has such a high expression of complement factor C3? What's the biological meaning of this?
Highly esteemed opponent, if I get you correctly, you mean why adipose tissue has more associated with a complement C3, right?
I don't know what's the biological function of the high expression of complement factor C3 in adipose tissue. Why does it need this complement factor?
In generally, the main source for complement C3 is the liver. We think most C3 is from liver, and adipose tissue is also another source for complement C3. In adipose tissue, it can produce complement C3, and in adipose tissue, there are not only adipocytes, they also include the immune system, and the immune system also can produce complement C3.
Okay, but I think that what we found was that adipocytes themselves really also express the complement factor C3. So can you envision a local function for C3 in these adipocytes?
You mean the local C3 the local effect of C3 on adipocytes?
Yes. Yes.
Yes, weight loss could produce. Adipose tissue could produce complement C3, and this is one way. And in another way, C3 may contribute to differentiation for the adipocytes.
Okay, so you think it plays a role in differentiation of adipocytes. Okay, that's an interesting thought. And I would like to continue by exchanging your thoughts about proposition number 5. Could I ask one of your paranyms to read proposition 5?
Complement factors have potential in risk prediction for obesity-associated cardiometabolic diseases and may also be the potential targets in treatment of obesity-associated cardiometabolic diseases.
Okay, thank you. What I would like to ask you is, what is your favorite complement factor that you would envision being a target for intervention? So what complement factor should you make a drug for?
So you mean which complement is the main driver for the association? Right?
Yes, you talk about potential targets. What target are you thinking of? C3, D, C3A?
And in our analysis, we have 3 complement factors: complement factor D, complement C3A, complement factor D, complement C3, and complement C3A. We found for the cardiovascular disease, complement factor D could be a main driver. And for the type 2 diabetes, complement C3 could be main driver because complement factor D, complement C3 could be a main driver for the type 2 diabetes as outcome because this it could be one mediator. And for type 2 diabetes, complement factor D could be a suppressor because complement factor D protective people with type 2 diabetes, and C3 can take a little bit effect on the association between obesity and type 2 diabetes.
Okay, so if I understand you correctly, you would like to suppress factor C3 and stimulate factor D, so factor D for cardiovascular disease, for atherothrombosis, and factor C3 for cardiometabolic disease, but specifically type 2 diabetes. Is that correct?
Yes, I think so. I think for the type cardiovascular disease, factor D could be the main driver, and for type 2 diabetes, complement C3 could be the main driver.
Okay, so thank you for those thoughts. And then take it one step further. How would you envision then targeting, for example, factor C3 in type 2 diabetes? And at what stage do we need to do this?
You mean how to explain the association between C3 and type 2 diabetes?
No, how do you envision targeting complement factor C3? Do we need an antisense molecule, or do we need an antibody to basically downregulate C3? So what were your thoughts about that?
You mean, if I get you correctly, you mean how to target on complement C3 and to treat this kind of disease, right? Yeah. And for treat this kind of disease, if we target on complement C3, we could use complement C3 antibody. In several publications, they have already used complement C3 antibody to treat one disease in our one kind of a disease in our mouse. This is one inflammatory disease in mouse. In there, complement C3 are higher than normal people. They use the complement C3 antibody to treat that disease for a short time. After that, the disease after that, the disease was cured, and they also found the risk for type 2. They also found the risk for cardiovascular disease is less.
Okay, I thank you for these answers, and that's enlightening. I think I have to give the word back to the orator.
Indeed, thank you very much. We need to proceed. So the opposition will be continued by Dr. Koppelijn. We move now from Leiden to Groningen. She is affiliated with the Department of Lifestyle Epidemiology, also a member of your assessment committee, and I'm pleased that she likes to join this morning, and I'd like to give her the word. Dr. Koppelijn.
Thank you very much, Mr. Rector. Dear candidate, I want to congratulate you and your team with this interesting thesis. And I also noticed that you use people first language in your work and also in your presentation that you just gave to us. For example, you refer to people living with obesity or people who have type 2 diabetes rather than calling them diabetics. This is very respectful language. And as the chair of the Dutch Association for the Study of Obesity, I highly encourage the use of people first language. And I would also like to ask the audience to take notice of this. So thank you for that. And it was also very nice that you explained the complement system clearly in your introduction to refresh my memory. What I found most intriguing is that you explain about the potential dual effect of factor 3, C3A, and factor D, like Professor Rennenberg before me already said. On the one hand, it could have protective effect on beta cell function, and at the same time, it may be related to perhaps fatty liver or hyperlipidemia. And that is what I want to ask a question about in chapter 4. In this chapter, you test the hypothesis that the association between adiposity and impairment in carbohydrate metabolism is related to changes in the complement system. However, you do not really look at this dual action for C3A or adipsin. You did not look, for example, at beta cell function. My question is, why did you not look at that? Because it seems to be a strong motivation to investigate the complement system in the first place.
Esteemed opponent, thanks for your question. Thanks for your kind words, and thanks for your question. And in chapter 4, we look at the potential mediation effect of complement system in the association between obesity and type 2 diabetes. In there, we have several outcomes: is insulin resistance and fasting glucose and the prevalence of type 2 diabetes. We we did not include beta cell function into our analysis. We have several reasons. One reason is now we have so many predictors and so many outcomes and so many mediators. There's a lot of workload. If we include a beta cell function in this analysis, it will be too much. This is one reason. Another reason, for beta cell function, there are several measures, not only one. It's not easy to pick out one perfect measures. So we did not include a beta cell function into our analysis, and we may, in the next step, we will look into that in our next chapter or next step.
Yeah. And if you do so, what do you expect to find?
You mean for the beta cell function?
Yes.
And because we found, because in the publication they show a complement factor D and has a could protective beta cell function and beta cell survival. So we expect complement factor. We expect there will be a suppress between the complement factor D and the association between obesity and beta cell function.
Right. And also to get back to the question of previous question, what is the reason that beta cell function is being preserved in response to complement activation? Is this a natural function, or is it related to pathology?
Thanks for your question. And for this part, until now, I do not have a perfect explanation for that.
Do you have some idea of your own, maybe not your research, but a hypothesis?
Sorry, I just get the impression for factor D could contribute to beta cell function and survival. I do not know very much for the behind the reason.
Because my next question is, I wanted to ask you if factor D is really an adipokine. So it is maybe produced by the adipocyte, or like you say before, perhaps by macrophages in the adipose tissue. But do you think it is really an adipokine? And the reason for that is that factor D and also C3A are not so strongly related to saturated, visceral adipose tissue or subcutaneous adipose tissue in chapter 4. And if the betas, they are all standardized betas, is that correct?
Yes,
Sentence supplemental table S2 on page 116. And they are all standardized betas, right?
Yes, they are all standardized.
So we can make the interpretation like it is a correlation coefficient, which would mean that the associations are weak to very weak if you correlate factor D to visceral adipose tissue or subcutaneous adipose tissue. So if this correlation is known, is it then an adipokine? And as an medical
I'm
client,
sorry to interrupt, otherwise we get too long. You can have a short question and then I give you a short answer, but not repeating question.
okay. So is it related in cross-organ talk?
Yes, in this chapter we standardized the complement factors, and in the results we found, we think complement factor D is adipokines, and because complement factor D are mainly from the adipose tissue, and the C3 complement factor D mainly from adipose tissue, and it also from the adipocytes and macrophages and other resource. And for C3, it also come from the adipocytes and the immune system. It just look at the beta factor D is not very high. It's less than C3, and C3A is less than C3 and factor D. This may be because the concentration C3 has much more concentration than complement factor D.
Yeah. Thank you. I give the word back to the rector.
Thank you very much. Then the opposition will be swiftly continued by Dr. Rensen. He is affiliated with the Department of Metabolism and Cancer Casisia from Maastricht University, a member of the assessment committee, and I'd like to give him the word.
Thank you very much. So first of all, I also would like to congratulate you and your team with this really nice piece of work. I really like the complement system, so I enjoyed reading it a lot. And I also really appreciated all the statistical approaches that you did. It seems to be very thorough. Also in the discussion section, I really noticed all the pitfalls and the biases that could be potentially present here. So it seems like you know a lot about this, much more than I do. So I will not ask about that, but rather zoom out a little bit and ask you a question about the complement system and the ways in which C3 and C3A can be generated. And you already showed that there's two other pathways besides the alternative pathway, the classical and the lectin pathway, so I won't have to ask about that. But I would like to ask you about the differences between these two pathways and the alternative pathway that was the main focus of your study. So what is the most important difference between the two other pathways and the alternative pathway? And what is the same actually in those pathways?
Esteemed opponent, thanks for your question. Complement system has three pathways: classical pathway, lectin pathway, and the alternative pathway. In my analysis, I focus on the alternative pathway, and the alternative pathway is spontaneously activated, and for the other two pathways, they are activated by initiator. They can't be activated by themselves or spontaneously. That is the difference, and that is one difference. And also for other difference, in our previous colleague study the alternative pathway, alternative pathway, lectin pathway, and classical pathway in one independent cohort. They found the classical pathway could be do the protective effect and harmful effect to cardiovascular disease. Also for the lectin pathway, they also can provide the complex effect for cardiovascular disease. And for the alternative pathway, they they provide the harmful effect to the cardiovascular disease. And for the same thing, they have all of them have the same complement factor, have some same complement factor.
Yes, thanks for this nice summary. So there's differences and similarities. You also said that indeed classical and lectin pathways have also been shown to be involved. But still, in your work, you focused on alternative pathway, you say. And I was wondering, because you basically measure C3 and C3A, and these factors can also result from activation of the other pathways, right? So how do you know that in your studies, it's really the alternative pathway that is important?
And in our analysis, as what you said, complement C3 and C3A may come from other alternative pathway or may contribute to other complement or may contribute to other complement. Because alternative complement pathway could be activated spontaneously. If one people without the initiator for the other two complement pathway, then the C3 and the C3, we can say the C3 and the C3A are only from the alternative pathway. Another reason, and complement C3 and C3A are mainly from the alternative pathway because the alternative complement pathway is activated via the C3 combined with water, spontaneously combined with water. Yeah.
Okay, so you think you show indeed that because of factor D, and it's a rate-limiting enzyme also, you mention a lot in your thesis, that this could be the most important pathway. I can understand that. I still would like to ask you if you think it is important to know which pathway is the trigger for the complement activation in cardiovascular disease. Would it be important to know this? In your opinion?
In my opinion, according to the previous publication, all of these pathways can work on the cardiovascular disease, and the other two can work on the back effect, and the other two can protect the other two can provide the bad effect and the protective effect. And for complement factor, for this company, for the alternative complement pathway, they only provide the bad effect. So I think it's, if we focus on all the complement, it's good to tell them. If we just focus on one complement, I think our analysis is good.
Okay, thank you. I don't know if there's no time anymore. Okay, thank you for your answers. I give the word back.
Okay.
Thank you very much. Then the opposition will be continued by Professor Dr. Li. He's affiliated with the Department of Nutrition Epidemiology of our university. I'd like to give him the word.
Thank you, Mr. Chairman. Dear candidate, your thesis is about complement, but I want to give you a compliment about your very nice presentation and the thorough work you did writing this thesis. Of course, I do have some questions as well, and I want to take you to chapter 5. And my first question is, in the intervention study, you do not mention about the participants, and I would be interested to know that. Were they checked for taking supplements, for nutritional supplements? Because, for instance, there is indication that N3 fatty acids from fish oil, they have effect on complement, they lower complement. So I wondered whether this was taken into account. So was it checked whether they took supplements?
highly esteemed opponent, thanks for your question, but I didn't get you very well. Could you just repeat your question?
Was it checked, was it verified whether the participants of the intervention study, whether they took supplements or not? In the beginning?
According to the weight loss study is designed by our colleague to study their study aims, and just according to the protocol, the people should just take the food as we provide.
Yes, but that was during the study. But before the study, did they take supplements?
And we do not check that. Yeah.
You did not check.
Yeah,
okay.
Yeah.
Okay.
I think we don't.
And second question is, if you come to table 1 on page 140, and there's a comparison of the baseline characteristics of the population. That's a very nice table between the lean and the obese subjects. But in the intervention study, there were only the obese subjects, right? Now, usually, you check the data at baseline, whether the randomized groups, the intervention group and the control group, whether were comparable. Did you check that, and were they comparable? Because I missed that in the table.
In the table 1, we check whether there is difference between the lean and obese people.
Yes.
And in the table, wait a moment, I remember we did it. In the table, in table 2, we check the difference between complement factor D3 and C3A after, and we check if the weight loss influence the complement concentration, and also in the supplement table. Also in the page 155, we check whether complement are different in the lean people and obese people, and whether they are different in the control group and intervention group.
Yes, I have seen that, and that is very nice information. But my question was, for instance, were the participants were randomized to the intervention group and the control group? Were those comparable in weight and waist circumference, in BMI, and so on, all the factors you mentioned in table 1? Were they comparable between the intervention group and control group?
For the and for this, for BMI. Thanks for your question. And for the study design, we first divide the people, the obese people, into 2 age groups. Then we randomly divide them into the weight stable group and in the weight loss group. Then we do not check whether they are same or difference between the 2 groups.
Okay, because it may be that, for instance, just by chance, you get more higher weight in the intervention group than in the control group, or the opposite. Okay, thank you. And the last question I have...
Sorry, there is no last question.
Okay, yeah, thank you.
Because we have a last opponent, and I would like to give also the opportunity to the last opponent to discuss with you. So we have Dr. Damen. She's affiliated with the Department of Immunology at our university, and I'd like to give her the final moment for discussion.
Dear candidate, first of all, congratulations on your thesis, also to your supervision team. I enjoyed reading it. I actually learned a lot about the possible roles of complement in cardiometabolic disease. I do have some questions about chapter 5. So my first question relates to the data or your analysis on intrahepatic lipid. As you find that lowering of plasma C3 is with weight loss, it's not mediated by changes in intrahepatic lipid, and thereby you kind of state that the liver might not be involved there. So my question is, since I would say liver pathology is not only reflected by liver lipid, whether it could be that the changes in liver fat content simply don't reflect changes in liver C3 production. So that so my question is whether you really think that the liver is not involved.
Esteemed opponent, thanks for your question. In our analysis, we found the reduction of complement C3 could be explained by visceral adipose tissue. In our analysis, we focus on the weight loss on the different adipose tissue and to check if the different adipose tissue could explain the effect of weight loss on complement concentration. And generally speaking, because complement C3 is mainly from liver, so we may think liver may involve in this process. And just from our results, we found it doesn't involve in this process. Because we have the intervention period is around 2 weeks. I think may these 2 months. May these 2 months weight loss intervention doesn't influence liver function, and if we think liver weight loss can influence liver, it will be via the inflammation. But in our analysis, we do not find weight loss influence the low-grade inflammation. So I think they may be not.
Okay, so you think the liver may not be involved, but would there be any other parameters that you could have measured and included in your analysis to check this?
Because the liver function have several measures, then the liver fat is one measure of them, one measure of liver function. Then for next step, we may include other measures for the liver function.
Can you name any or? Okay. Then next question. So related to that, you hypothesize that lowering of C3 with weight loss is caused by the reduced production in the visceral adipose tissue. So what follow-up studies would you perform to look at this, to confirm this?
Yeah, thanks for your question. And in our analysis, we found C3 the effect of weight loss on complement C3 is via the reduction of visceral adipose tissue. Next, if we confirm this, we could use we want to first measure the gene expression, the C3 gene expression in adipose tissue. If the gene expression is less, then they can confirm this.
Okay, so then you mean in humans, in the gene expression?
Yeah.
Okay, so would there be, would you also perform any animal studies or mechanistic studies?
If in animal study, and we also can measure the gene expression, and we can measure the C3 gene expression in the animal study, and we can make the C3 gene knockout. Then we will then we will look at the outcome, what will happen.
Okay, thank you. So then my next question relates to sex differences. So the study have been performed in men, the weight loss study. But you know, throughout your thesis, you do see some differences in the complement system and the outcome parameters between males and females. So do you think you would find the similar effects in women if you would perform this weight loss study?
Thanks for your question. And in weight loss study, we only include men in this analysis. If we
Dear candidate, you can finish your answer if you want. If you said, I don't want to finish, that's also fine. It's up to you.
Okay, maybe I can finish it after this meeting, after this defense.
Okay, you finish later. Dear candidate, dear Ms. Jin, the time appointed for defending your thesis has now passed. The degree committee will now withdraw to discuss the quality of your thesis and in particular the way you have defended your thesis this morning. I request that you and your company here present await the results of our deliberations and our return to the aula. Thank you very much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. fly by now, don't waste all your time. Cause I'll go the extra mile.
Dear candidate, dear Ms. Jin, the degree committee here present has assessed not so much the quality of your thesis, because that has been approved before, but in particular the way you have defended this thesis, your thesis, this morning. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor van Grevenbroek is authorized to confer upon you this academic distinction in accordance with Dutch university custom, and I'm pleased to invite your supervisor now to take the floor.
Please all rise. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Shunxin Jin, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Please be seated.
Dear Dr. Jin, congratulations on this achievement, a job very well done. Dear Shunxin, the first time we met was in December 2017 via Skype. After that, we had some email exchanges and you applied for the scholarship, and in October 2018, you came to Maastricht. That must have been quite a change from the Beijing area where you were living at that time. The first year and a half of your PhD project, you work in the lab, measuring the complement factors that are the basis of your thesis. You did these analyses partly by yourself and partly with the help of Vicky, because it was a seriously large number of measurements. You worked very precisely and obtained valuable data, although you did scare Mayu every now and then with your sudden jumps and movements in the lab. Besides working on the complement measurements, you also worked hard to get acquainted with epidemiology, especially with the analysis of observational studies. This was a new field for you, but you studied consistently and you got to know and understand the background, the logic, and the lines of reasoning that are used in this discipline. In the summer of 2019, we had our lab day out in the Gaia Zoo. I remember that you picked some fruits from a tree and ate them. At that time, I was not so sure that was a good idea, but you were clearly better capable than me to distinguish edible from unedible fruits. Later, I found out that your family grows cherries, so that explained a lot. You can recognize a cherry tree when you see one. In September 2019, you and other members of the research team went to Barcelona for the yearly symposium of the European Association for the Study of Diabetes. And the visit to Barcelona was nice, and you did a good job presenting the poster. A couple of months later, the first virus struck. It was a computer virus that hijacked our university, and after that, you and all other UM employees could not access our data for a while. That did hamper your project. And we were not yet aware of what would come next. Because when all that was solved, it was early 2020, and the second virus struck. This time it was a biological, it was worldwide, and it had a very large impact, not only on our work, but also on our lives. And with two viruses within your PhD studies, I must say, it's a really good thing that we are epidemiologists and we know that association is certainly not causation. And I really admire you for not giving up then, when you were mostly or completely had to work from home for almost a year and a half. Just you and your laptop and an occasional face or faces on the screen. I have come to know you as a person who is genuinely interested in other people and cultures, and it's a pity that the lockdown hindered you to explore Europe more broadly. I was happy that in the late summer of 2022, you did have the chance to go to meetings in Bern in Switzerland and to Stockholm in Sweden. In that last meeting, you got yourself in a situation that was particularly stressful for you, so that turned out to be not really relaxing after all. In the first years of your PhD, you struggled with English language, and the virus and lockdowns did, of course, not help in that regard. For almost 2 years, we had meetings via Zoom. We did not go to conferences, apart from the online ones with very limited interactions, and we could not freely travel around in the Netherlands and in Europe. But you did look for and found ways to improve your language skills. You used an app to improve your pronunciation and to broaden your vocabulary. And in our weekly meetings, you really actively looked for opportunities to include those new words and expressions in your sentences. And that really paid off. Dear Shunxin, you and I have very different relationships with deadlines, and particularly with sticking to deadlines. But I will not elaborate on that today. I have been chasing you quite a bit over the last couple of years, but that will stop now. You obtained your degree, and I am sure that you are fully equipped to continue running on your own. I wish you all the best in your future career. And with this, I want to close by saying that I hope that now all your worries and stress about this day will disappear in the blink of an eye, and that you are ready to celebrate this joyful day.
Thank you.
Yes. Dear Dr. Jin, how does that feel? Feels good?
Quite good.
Quite good. Okay, quite good. Also, on behalf of Maastricht University, I would like to congratulate you. Yes, this is an important day for you, and it is also the highest distinction which you can receive in the Netherlands at a university. So it's really important. I would like to congratulate you, but also not you, but also your promotion team, because you work with them, I heard now, for the last 5 years, and they basically brought you in these 5 years to this day where you could today, where you have been shining. I also would like to thank your promotion team that they help you. I also would like to thank the members of the Corona, the 6 members who have questions to you today. Maybe they were a little bit tough to you, the questions, I'm not sure, but I think you answered them quite correctly. I also would like to congratulate your family. Maybe they are online at the moment. Are they looking also today? You don't know?
They are not here. Yeah.
Anyway, it is recorded, so they can look later to what you have achieved today. I would like to address also the other CSC students here in the audience. I mean, you saw this morning how one of your colleagues is defending a thesis defense. I know there will be several coming in the next months and years, and I'm looking forward as a prorector to guide them also to this thesis defense. By this, I'm going to close this session, and for you students and your members, I have a hammer here, you haven't seen it this, and with the hammer, I close this session. And how do we proceed now? It's the following. There will be a reception. And we make a separate picture on the stairs here of our university. And I hope that we will meet you there in about 5 to 10 minutes. Thank you very much.